All right. Well, as Annette said, thank you very much for being here. My name is Anne-Marie Foulbrood, and I'm the Tech Transfer Team Coordinator. And so what I'd like to do this morning is I'd like to introduce you briefly to the Be Informed Partnership and the Tech Transfer Team program. As some of you may know, we are a small but impactful nonprofit organization that is spread throughout the country. Our reach is fairly broad, and we have a big extended family. We work with many collaborators from all sides of the industry, specifically closely with over 10 universities across the US. We have about 12 to 15 full-time staff members and a handful of graduate and undergraduate students either working with BIP data or working in the lab helping to process samples. Our main mission is to improve colony health, and we do this by supporting beekeepers, bridging the gap between science and industry. And in the process, we became the largest repository for colony health data in the United States. So BIP is doing a great many things, and some of you may be familiar with some of our work. We offer a variety of programs from different types of beekeepers, from commercial to sideline to hobbyist. In our first webinar, we shared information about the National Colony Loss and Management Survey. And just a few weeks ago, we had a webinar featuring our Sentinel Apiary program. Today, the Be Informed Partnership Tech Transfer Team Honeybee Health Specialist will guide you through causes, signs, prevention, and treatment of a few common honeybee brood diseases. You may be wondering what qualifies them to present on this topic. So let me introduce you to the Technical Transfer Team program and the work that the field specialists do. The tech team is spread across the country, and on this map, you can appreciate the territory covered by the program. The yellow dots represents the home base for each of the BIP tech team field specialists and the university that they are affiliated with. And then you have the orange dots, which represent all the states in which the tech team program provides services in. And then the arrows are meant to represent the movement of the beekeepers and therefore the movement of the tech team that follow their beekeepers' honeybee colonies. And finally, the blue dot is the diagnostic lab at the University of Maryland, where the samples are sent to be processed. So in essence, our team has location and reach in each of the big US commercial beekeeping hub, and they follow beekeepers' migratory routes. So basically, we go where the bees go. In summary, we are based in 5 states. We perform services in 17 states. 10 of these states are the top honey producing states in the country. So the tech team program works with over 110 commercial beekeepers, and together these beekeepers manage over 500,000 colonies in total, and therefore represent approximately 17% of the total honeybee colonies in the US. So we call the tech transfer team field specialists or boots on the ground. They perform full colony inspections, and they also record any overt signs of diseases. Any samples, and they sample for a variety of tests, including Varroa, Nosema, along with viral and pesticide loads on demand. They also perform hygienic testing, mainly with the California queen breeders, and they perform research trials. So in numbers, this represents in 2019, they collected about over 11,000 samples. They performed 3 trials, and together they traveled over 100,000 miles in their BIP trucks. So that's a lot of territory covered. So considering the wide variety of commercial operations that they visit and the number of colonies they inspect in depth every year, the field specialists have accumulated a wealth of information, and especially regarding diseases. So we decided to break up the disease topic into a few chunks. We plan to offer a webinar solely on Varroa and another one on adult bee diseases in the weeks to come to complement today's offering. But today we will mainly focus on brood diseases, specifically addressing American Foulbrood with Dan Wines from Michigan, European Foulbrood with Rob Snyder from California, Chalkbrood with Nelson Williams from Minnesota, and finally Parasitic Mite syndrome with Ben Salmon from Oregon State University. So without further ado, let's get started with our webinar and our second poll. Take it away, Nat.
Okay, hopefully you can see the poll on your screen. Please go ahead and select your answer. Going to give it another 15 seconds. Okay, I'm closing it up so we can keep moving. I'm going to share the results. Okay, I'm going to close down the poll and you guys can keep moving.
I guess you are up. Dan Wines from Michigan.
So looked like we had most folks were seeing that American Foulbrood, kind of the most serious, but thankfully relatively uncommon these days. It is a bacterial brood disease, Bacillus larvae. You kind of see, we'll get into more of the visual signs, but this kind of close up on some brood here shows a few of the things you'll see in an infected colony. One thing at the top to note, it is in some jurisdictions, it is a notifiable disease. It means if you do find it in one of your colonies, you may be obligated to report it and follow very particular regulations as far as how it's dealt with. But that is largely on a state-by-state basis. So as a beekeeper, it is your obligation to know, local and state beekeeping laws, and particularly as they pertain to American Foulbrood. As a disease, it is generally fatal if the beekeeper does not intervene. That's kind of why we consider it the most serious of the diseases. There's persistent spores. It's a bacteria that forms spores, and they remain viable for decades. It's extremely infectious. It only takes something on the order of about 10 spores to infect a larvae. And at the end, after the bacteria has fully consumed the larvae, it produces millions of spores. So its kind of magnification is very high. And that's why it's, it is serious. It needs to be taken seriously and dealt with. It's pretty, because of these spores being persistent in hive material, it's pretty easily spread through colonies just through normal beekeeping work, moving from hive to hive with the hive tool, hands, gloves, things like that, or just beekeeping drift. So just through routine basic operations, it can be spread. So we can go to the next slide and we'll look a little bit here about the biology of it. I said it is a spore forming bacteria. The infection occurs when a young larvae gets fed infected brood food. The spores germinate inside the larval midgut. The gut lining then eventually ruptures and the infection spreads to all of the tissues in the larvae. Death typically occurs right around the point of capping, just as it's transitioning from larvae to pupa, kind of in that pre-pupa stage, give or take. There's a little variation in timing, but generally speaking, it's right at the point of capping. The larval breakdown continues after this point, and this is when kind of the bacteria transitions from its vegetative state to spore formation. And the adult bees can carry the spores on them, but they're not impacted. So that's why we say it's a brood disease. It doesn't impact the adult bees, but they are carriers. They can potentially be vectors of those spores within the hive, but also to other hives if it's foraging, drift robbing, things like that. So we can move on to the next slide. And that will start to look at kind of the recognition and diagnosis here. As you're opening your colony, what are you going to be looking for? Initial infection of a colony is just going to be present in a couple cells. One of the things beekeepers talk about with Foulbrood is a smell. It will, as it progresses and there's a lot of larval death, there will be kind of a general smell of decay. I've heard it described a lot of different ways, and because of that, I don't really like to use it as a diagnostic because I've heard it described 10 different ways, and everybody's sensory perception is different. So the smell is not really a reliable diagnostic, but if you do smell something off, it's just kind of a clue to be looking for there, a little extra vigilance. But as the infestation progresses or infection, the population of the colony is going to dwindle. You're going to see a very spotty brood pattern. We kind of see a little bit of that here, where it's very shotgun looking. There's mixed age classes amongst the brood. The two primary things that I look for, these kind of highlighted in bold here, are the sunken perforated cappings. Sometimes they look greasy or wet. They've got holes chewed in them. As you can see, there's a variety here circled in red that just kind of show this kind of torn look. Those, as you're doing a brood inspection as a beekeeper, that should really get your attention. You want to look closer at those cells. The other thing that really catches my eye and points me toward American Foulbrood is kind of this melted, slumped, just kind of a brown, smooth material. It's kind of got a silky, liquidy texture to it. Get a caramel color, latte color. So if you're seeing that, these two things that are kind of highlighted there, those are things you're really going to want to pay attention to and look further. We'll talk about how to do some testing here in a few minutes. And then as the disease progresses further, as these kind of liquid, they dry down to a scale, which can be quite hard to see. We have some photos coming up here in a minute that'll kind of show you what that looks like, but it's generally not the first thing you're going to see. And once a colony has a lot of scale, it's progressed fairly far in the disease. Another thing to talk about is the pupal tongue, and we'll show some pictures. That is a visual sign that is unique to American Foulbrood, but not often present. So not the primary diagnostic, but we do have some pictures of it that we'll explain. So we can go to the next slide and we'll see some more kind of close up look at some of these pictures. Again, on the left here, just kind of a shot of a pretty good sized chunk of brood, and you just see a lot of different things present here. We see torn cappings, we see sunken cappings. There's a lot of kind of greasy looking or moisture condensation as these larvae are breaking down under the capping. Some are getting opened up, some haven't been opened up yet, but that's, if you see that in a colony, that's a very advanced infection that's really progressed. Here we kind of see on the right, a close up in the texture of what you're looking at there. It's just kind of melted down into a light brown, caramel colored sort of liquid goo. It's got a little viscosity to it, but it's pretty thin. It's not generally very gummy or dry. We can move on to the next few pictures. And again, this one is further drying on the left here. It's kind of zoomed in. It's not quite a scale yet, but it's getting very close to it. Its color's darkened a little, it's sunk further. And then the photo on the right, we see the scale. And on the bottom of the cells, now this is, as a beekeeper, if you're inspecting for scale and you hold the frame as you normally would for a brood inspection, you're probably not going to see it because it is down kind of flush on the floor of the cells. So you want to hold the frame kind of with the bottom away from you, so you're looking and you can see the bottom of the cells, and you'll get kind of a glint of those scales. So that's something to look for. Generally, if you've got a colony with scales, you're going to see these other symptoms, and I personally find it much easier to see the torn, sunken cappings and the kind of melted larvae. But this is something to be aware of, particularly in old equipment, used equipment, inspecting dead outs and things like that, where there's not a lot of brood to look at. Looking for scale in the cell bottoms is an important thing to do. So we can move on, and we will look at, this is one I mentioned, the pupal tongue. This one gets a lot of confusion, and it's the reason I put up both of these pictures. So this picture on the left is the pupal tongue. That is a pupa that's succumbed to American Foulbrood. It's got that characteristic color, and that vertical little kind of triangle at the top there is the proboscis, the tongue. Not very common. Why I wanted to include it is get a lot of people saying, oh, I have dead bees with their tongues out. That's AFB. That is not AFB. This picture on the right is bees that died from another cause. In this picture, it was pesticides, but bees die looking like this from, could be starvation, could be chilled, neglected in a dwindling colony. So when you see an adult bee, fully formed head and things like that, that dies with its tongue out, that can indicate any number of things, but it is not indicative of American Foulbrood. So just to distinguish between those two, tongues out does not mean AFB. People tongues a very distinct visual presentation there. So now, as far as we've talked about the visual, we can move on and kind of, say a lot of the diagnosis of this is just going to be eyeball inspection. Thankfully, it is one that we can see. Moving to kind of confirming what we think we found. The simplest, most common way I usually test for, I find this to be an easy and reliable test, is we call it the ropey test. You'll hear it called matchstick test, ropiness, things like that. Basically, you take one of these and suspected infected cells with this brown kind of silky liquid in it, and you're going to poke a matchstick, a twig, in this case, a popsicle stick, coffee stick, something wooden, something that's not real slippery, so this liquid can adhere. And then if you draw it out just gently, it'll rope out in a nice smooth string like that. It should rope out to at least 2 centimeters, so nearly an inch. Sometimes you'll get other larval death, and you'll have kind of a liquidy substance that'll rope a little, but it won't get anywhere near this length or the smoothness. So if you poke something in and bring it out and it ropes out to near an inch, that's a pretty confident diagnosis of American Foulbrood. So that's one I like to use. We can move on to the next. There is now kind of an upgraded capability. In the last handful of years, these test kits have become fairly widely available at a variety of beekeeping supply companies. Most anyone carries them. They make them for both AFB and EFB. They are separate kits, so depending on what you're trying to confirm, but it can be a pretty good thing as a beekeeper to have at your disposal. They're pretty reasonably priced. They typically last for about 1.5 years or 2 years when I get them. That's how long for the expiration date. So most similar, you're going to find again that suspect larvae. You scoop it out. This kit contains everything, including instructions. So this is a simplified, kind of explanation. But essentially, you scoop out a suspect larvae, put it in a little solution bottle with a stainless steel agitator BB, shake it around for 20 seconds. They include a little pipette, and you put a drop of the resulting liquid on the circular well of this test plate, and it takes 20 to 30 seconds. The moisture will kind of seep across that rectangular viewing window, and that will tell you the result. You should always get at least one of the bars on the bottom. You see there that one bar, that's the control bar that indicates it's a valid test, and it's reliable. On the top, if you see that second bar that's filled in at the left, that indicates that's your test strip or your test indicator, and there with the 2 bars, that indicates a positive test. Even if it's faint, it is. If you see any hint of a second bar there, it is a positive test. You have confirmed American Foulbrood. So I like these. I use them when in doubt. There is one more method available. We can move on, and that is just to say the USDA Bee Research Lab in Beltsville, Maryland will test samples. I didn't bother writing in the website here because it's quite an extensive URL. But if you just search USDA Beltsville Bee Lab, BRL for Bee Research Lab, you'll find their website, services available. They have sampling and shipping instructions. It's all there. They will, again, test for EFB and AFB. It does tend to take a little while, one just with the shipping and getting it to them, and then depending on their workload and how long. So it's not something I typically do for AFB because of the seriousness of it. I feel like it needs to be dealt with immediately. I don't want to wait a week or more on a result. I want to know when I want to deal with it. One nice feature of this, though, that the Bee Research Lab does do is they will test for antibiotic resistance if they do find the sample you send is positive for AFB. So that is nice. It is something we'll talk a little bit about antibiotics here in a minute. But resistance is a concern. There is some AFB that has been found resistant to antibiotics. So that service is available. Now that we've kind of talked about the diagnostic part of it, we can move on to kind of response and treatment if you do confirm a colony with American Foulbrood. Couple things you want to do. First, we'll talk about what you want to do at the colony level. And again, this may be, you may not have a choice in this. This may be regulated depending on where physically your bees are, what state, and the regulations within that state. So you want to know that. First, and the thing I do and have done is to burn the colonies. It's not pleasant, but we've talked about how contagious this disease is, and I'm of the mindset of better to burn one today than a dozen tomorrow. It does spread. It is a problem. It does not go away on its own. So that is one method. There's lots of instructions on how to do this if you find yourself in this scenario. Honey does not burn particularly well, so typically you want to burn in a hole and then bury a couple feet deep so there's not remnant honey that's going to get robbed out. Anything that could contain spores, if it's not completely burned to ash, needs to be buried. Another method is beekeepers call it shook swarm or frame exchange. This basically involves shaking the bees off onto brand new comb and feeding them, and then feeding them antibiotics, and then again still burning that contaminated equipment. You're not going to be able to sterilize frames and things like that. So you do want to, with the infected colony, that is another option to kind of save the bees, destroy the equipment. Not going to be a viable option late in the season. That colony still got to have enough time to build up and make some strength. So depending on where you're at and what you may be allowed to do and what time of year, you have a couple different options. So that's what you want to do with an infested colony. This next slide talks about at the apiary level. If you found AFB in any colony in an apiary, that apiary kind of becomes now a red flag. You want to treat every colony in that yard with antibiotics. You want to, any equipment or something that may have had contact wants to be sterilized. You can do that with fire. Generally, at large scale, things can get sent for irradiation. Boxes can get wax dipped. They do need about a 10-minute submersion at 160 Celsius. So pretty serious thing we're trying to destroy here, and not to be taken lightly with the kind of the cleanup aspect of what you're going to do after the fact. But again, if you're destroying infected colonies and treating all those in the yard, and then you do want to kind of quarantine that infected apiary where you found it. Nothing, minimize what comes into the yard, certainly what goes out of the yard. You may want dedicated hive tools and things like that. But certainly no hive material, boxes, woodware, anything like that should be leaving that yard for probably a couple of years, until you've been through and inspected several times and had no further positive diagnosis. And then just to close here, because I have talked about antibiotics, and it's going to come up in the next section as well. As far as treating antibiotics, they do recently, as of about 3 years ago, they do require either a prescription from a veterinarian or a veterinary feed directive. As a beekeeper, you should have a relationship with a veterinarian. They will be able to provide you antibiotics when you do have a positive diagnosis for American Foulbrood and also European Foulbrood. There are 3 antibiotics approved for honeybees. I won't get into the particulars of that here. That's a little beyond the scope, but it is very important. They come in different mixes and concentrations. It is very important to apply according to the label, one, so we don't create or accelerate this resistance, and also so we don't potentially contaminate food supply, get it into honey. There are withholding periods beyond when you finish applying, and then before any honey that would be harvested as a crop should be taken. Also with AFB, they do not work against the spores. That's why destroying infected colonies is really the only option. They just halt replication in the infected form. So I will leave it there with American Foulbrood. We kind of started with one of the, not a good one. Not that any of these are great, but we will move on to some of the coworkers talking about some of these other diseases. I think we'll probably take questions at the end.
Okay, we have another poll up on your screen if you wouldn't mind filling it out. Another 10 seconds. All right, I'm going to end the poll and share the results here. And in the meantime, if Anne-Marie, if you could introduce our next speaker.
My apologies, can't unmute. So Rob Snyder, one of our 2 California Tech Transfer Team field specialists, is up next talking about European Foulbrood, and hopefully he will answer the question from the poll here. We are a little separated on the answers here, so that would be great. If you want to take it away, Rob. I cannot move until the poll is removed. Here we go.
So European Foulbrood is caused by the bacteria Melissococcus plutonius. And as Dan was talking about American Foulbrood, that's a spore-forming bacterium. And European Foulbrood doesn't cause spores, so it's not as hard to get rid of. It was discovered in Australia in the late 1970s, and now it can be found throughout the United States. And we're starting to see this disease much more prevalent, at least in California. And it's often found in early spring and in wet, cool environments. Also, sporadic nectar flows can cause this disease. And I chose this picture here to start off with because this is often my first indicator of what the disease looks like. I'm just looking at this spotty brood pattern, and this is a giveaway that something is wrong with the hive. And if it's this spotty, it's usually European Foulbrood or some of the other diseases. If we can go to the next slide here. So EFB biology is a little more harder to follow. So basically, you have your ingestion of spores, and they replicate in the midgut. And depending on the amount of spores that are ingested, the larva can die at different stages, which gives you all this variability as far as the symptoms. But I've kind of showed you here some images of some contorted larvae, and as they contort, eventually they'll sink. Or on the top left picture, you'll see the larva sunken all the way to the bottom of the cell with the defined trachea. But basically what the bacteria does is once it's ingested, it outcompetes the larva for food. And I'm going to show you some more of these different stages of European Foulbrood as far as visual symptoms. We can go to the next slide here. So you'll see with the green arrows, you have these contorted or twisted larvae. And then you'll sometimes see these C-shaped larvae, which are pointed out by the orange arrows. And then discolored brood food, I'll point that out on the next few slides. And then you'll see in the yellow arrows, you'll see defined trachea from the larva, these white lines. This is a really distinguishable thing to see with European Foulbrood. And then sometimes you'll see melted larvae where you see the red arrows pointing those out, and that's a result of different bacteria or the larva dying at a different stage in the head up position. And then also, as Dan said, you get an odor with European Foulbrood as well, but it's very variable, and often there's no odor. So I don't usually go by that unless I recognize it, from outside the hive before I even get into that hive. So next slide here. So here's some frames that were pulled out of a colony with European Foulbrood, and these were pulled out of the center of the brood nest, and definitely concerning if you're pulling frames of brood out and You're seeing this shotgun pattern where the brood's not all sealed together. And once you look closer, you'll start to see some of these other symptoms that I'm going to point out on the next few slides here. Next. So here are some of the early signs of European Foulbrood. I'm going to first go to the picture on the right with the yellow arrows pointing to this discolored brood food. This is not a really good way to identify European Foulbrood, but it is something to take note of, and you're going to want to check back on these colonies within a few days to see if the disease progresses. You'll see the green arrows on the bottom left-hand corner. Those are a little closer look at what you're seeing. The brood food's a little yellow. Sometimes it's brown. This could be due to the pollen source that's coming in. So it's not the best way to diagnose European Foulbrood. But when you look closer and you see that red arrow and that same picture, you'll start to see that C-shaped larva, and it's starting to look sunken. So that is concerning. And the picture above that with the orange circles around it, you'll see some of those cells have a little discolored brood food, and then also you'll start seeing these larvae in a C shape, and they kind of look a little deflated. So that's usually how I catch it early, is this C-shaped larva, and they look kind of flat to me. So that's one thing that I use to identify European Foulbrood early. We could go to the next slide here. We'll look at some of these signs in the late stages. So here you'll see with the orange arrows on the top picture, kind of that C-shaped larva and how it degrades and sinks down to the bottom of the cell. The red arrows on that same image are a little more discolored. They're probably a few days older, and there probably is some secondary bacteria starting to move in and change the color of the larva. And if you look at the image below, you can see with the orange arrows how you're starting to get more of this discoloration. You see different shades of brown. Sometimes you'll even see yellow. And then the red arrows on that same image is where you're going to get almost the EFB scale, which is very easy to remove, unlike American Foulbrood, which is pretty tough to remove without destroying the cell or parts of the frame. So these kind of just flake out, and they're really easily removed. And you'll see kind of on the right side on the bottom image is another picture of a larva that's deteriorated, and that'll soon dry out and turn to scale. And above there, you'll see some of these, what they call melted down or molten looking larvae. And there's also one that's kind of contorted in the top left on that right image. But once the disease gets to this stage and the larvae are this old, with the test kit, you're often not going to get a positive with the test kit, because there's other bacteria that moved in and have broken down the larva, and you're not going to turn up with a positive. And I'll talk about the test kit in a few slides here. So here is the test kit. It's pretty much identical to the test kit that Dan had talked about for American Foulbrood, and you pretty much do the exact same thing. One thing that I recommend with these test kits is I'll often pick 3 to 4 larvae as opposed to just one. And like I said, I pick these earlier stage larvae, which I have a picture on the next slide. And then you put these larvae inside the container and shake it for 20 to 30 seconds, and then put your drop of liquid into the circle on the left side of the test kit. And you're looking for the 2 lines, of course. If you only get one, it's probably not a positive. If you're still suspicious, you can always use another test kit. So we usually carry several in the truck in case we have a negative turn up, but we're pretty sure that disease is present. So here is a little expansion on the field test kit, and these orange arrows are larvae that I would pick for putting inside the test kit. If you see these yellow arrow looking larvae where they're discolored, brown looking, you know, just darker and not that pearly white, they tend to not give you a positive result in the test kit. So I always inform people to pick the younger larvae that look like they have symptoms of European Foulbrood, because often if you test the nastiest brown looking ones, you're not going to get a positive test result, and that's going to give you a misdiagnosis, and not treating this disease can run you into problems. And I've heard people having to deal with it all summer once it was in their operation, just from not having a proper diagnosis. So the only treatment that you can use for European Foulbrood is terramycin, and you do need a veterinary feed directive or to get this disease or get the antibiotic. Sorry, and one way you can get the veterinary feed directive is with image or the disease manual that we sell, and show the vet the pictures of what you're seeing, and then have one of those test kits that's positive. And I've heard that was a valid way to get the antibiotic prescription. So Tylosin, which is used for resistant strains of American Foulbrood, is not effective on European Foulbrood. So the terramycin will not work on, or not terramycin, the Tylosin will not work on European Foulbrood. And if you do have European Foulbrood, they recommend treating 3 times with terramycin about a week apart, and use the recommended label dosage. And I think that's all I have for European Foulbrood, and I'll answer any questions in the chat box.
We're going to move to poll 3 next. And you can, if you want to, Rob, start looking at some of these questions, but we will take questions at the end of the presentation so that we can do some of that. But you're welcome to look at the checkbox.
There is a real quick correction. I guess that something was annotated on a slide. And Natalie, could you speak to that? I'm wondering if Natalie can unmute. I might have to unmute for her. 1 second here.
Is it the spore thing?
Yeah.
Yeah. So Rob had mentioned that European Foulbrood does not end up forming spores. And then later on, he mentioned something about how the bees ingested the spores. He meant to say that the bees ingest the bacteria, and then they go and reproduce in the midgut. So EFB is not a spore forming bacteria. That's correct.
Okay, thank you. I just wanted to make that clarification since we're recording and that question came up during the thing. Okay, I'm going to go ahead and close the poll. It looks about 2/3 of us have responded. So let me end that and share the results here.
All right. So again, a little divided. Many are talking about parasitic mite syndrome. Where we're going to move into our next brood disease with Nelson Williams, and he will probably give you the answer to this question. Nelson, you are up.
I am here. Yeah, so I am Nelson Williams. I'm the newest tech team field specialist. I work in the Midwest in Minnesota, and I serve Minnesota and North Dakota beekeepers. And today I'm going to be talking about the causes, identification, and prevention of chalkbrood, which, to answer the poll question, is the most prevalent brood disease encountered by BIP field specialists. So next slide, please. Chalkbrood is a fungal disease caused by the fungus Ascosphaera aphis. The sexually produced ascospores are considered the primary source of the brood infection. So the spores germinate in the midgut of the honeybee larva, and as those spores germinate, the larvae rapidly reduce their consumption and then stop eating altogether. And as the fungal mycelium grows inside the larval body cavity of the bee, it eventually breaks out and leads to larval death, which is the result of the physical and biochemical damage. So colony infections of chalkbrood are the most prevalent during the spring, given that the fungal growth is enhanced in cool and humid colonies. The infection rates are directly related to the concentration of fungal spores in a colony. So the higher the concentration of spores, the higher the chance of infection. There are both biotic and abiotic stressors that can weaken the honeybee immune response, which is going to lead to increased vulnerability. So in the top right photo, you're seeing moldy frames, and the use of moldy frames can really weaken that immune response, leaving the colonies more susceptible. And then in that bottom right photo, you're seeing a damp and wet bottom board. And that can really add to the humidity in the colony, and that's going to enhance that fungal growth. So now that we've kind of covered the causes of chalkbrood, we can move on to the transmission of the fungus. And chalkbrood is a horizontally transmitted disease, meaning that it is not inherited or passed down from the queen to worker larva or drone larva. So the fungal spores are transferred from bee to bee or bee to larva, and they must be consumed by larvae 1 to 4 days old to exhibit signs of infection within the colony. Adults are not susceptible to the infection, but they can transmit spores within and between colonies. So the horizontal transmission of spores can occur a number of different ways, but a few examples would be the nurse bees feeding larvae, during mating flights, during foraging events, robbing, or through the use of spore-contaminated equipment. So hive tools, gloves, frames, boxes, all of these can be reservoirs for spores. We must rely on the visual identification of signs in the field to accurately diagnose chalkbrood. So one of the first things that I look for is the coloring of larvae in the cells. The infected larva's coloring has a similar appearance to brie cheese, which is that white and yellowing. So on the left, we're seeing that delicious brie. And on the right, we can see the white and yellowing of the infected larva. As we zoom out a little bit, we can still see the white and yellow coloring, as well as the drying out of the larva, which gives the infected brood its chalk-like appearance. And as we zoom out a little bit further, we can see the spotty brood pattern that generally accompanies chalkbrood infections. So once again, as Dan and Rob mentioned, the spotty brood pattern is not exclusively associated with chalkbrood, but rather a sign that closer inspection is necessary to determine the issue that's going on in your colony. So a spotty brood could point to EFB, it could point to AFB, it could point to chalk, or perhaps a drone layer or a failing queen, but you're going to need to look a little bit closer if you're seeing spotty brood. But on the left, we're seeing larvae that look very similar to the sackbrood virus. The only difference is the larval bee head is less defined, and there's a more rounded and sunken appearance. And the photo on the right, you can see infected larvae that have been pulled from their cells by tweezers. Just kind of as a side note, I think tweezers are really a great tool to keep in your beekeeping kit. to pull out and examine brood more closely. Just remember that you're sterilizing them between colonies and between apiaries. Next slide, please. Yeah, correct slide. Thank you. So always remember to check your bottom boards. As the infection spreads throughout the colony, Chalkbrood mummies that have been pulled from the cells will start to collect on the bottom boards. So on the left, we're seeing scattered Chalkbrood mummies on the moldy bottom board of a colony. And on the right, we can see an extreme case of infection taking on the appearance of a mass grave of mummies. So in cases of extreme infection, you will start to see Chalkbrood mummies spill out of the entrance onto the landing boards. So what we're seeing here is white, gray, and black mummies. And the color of the mummies is believed to be associated with the progression of the fungus. So the black mummies are the most contagious due to the advanced stage of mycelial growth and spore formation. And in these two photos here, we're just contrasting the early stages of Chalkbrood with the later stages of Chalkbrood. So the photo on the left, you have that classic appearance of chalk or brie in the cells. And on the right, as the disease progresses, we can see that drying and the color progression from gray to black as that the larva starts to shrivel to the floor of the cells. And this was just a cool photo that I wanted to include that Rob took. But this is a photo of a drone larva with a Chalkbrood infection. So Chalkbrood is not exclusively a worker brood disease, and it can infect all castes of brood. So workers, drones, and queens can be infected. So now that we know the signs of Chalkbrood, how do we prevent and respond to infections? Reducing transmission is going to be the best prevention. So remove old comb from colonies with spore contamination. When you cull out that old comb, you're really just getting rid of those reservoirs of spores. The spores can remain viable in comb for approximately 15 years, and freezing is not going to exterminate them. Make sure that you keep your bottom boards free of mummies. So when you're doing an in-depth inspection, make sure you remove that bottom box and scrape and sterilize your bottom boards. To sterilize your bottom boards, I think just a quick rag with a bleach solution will do a lot to clean up those spores that are forming on your bottom boards. Also, you're going to want to increase ventilation and airflow within your colonies. So increasing ventilation would include using screened bottom boards or drilling holes in the front of your boxes just to keep that airflow going. And finally, as a response to an infection, I would recommend that you requeen your colonies with hygienic queens. This will increase the likelihood of infected larvae being removed from the colony before the spores can multiply and spread the infection. And then finally, is there a treatment for Chalkbrood? To be determined. The antifungal properties of thymol that are used in miticides like Apiguard and Apolife VAR purport to be effective against Chalkbrood, but more research is necessary. So that's going to wrap up my portion of the presentation, and we're going to pass it along to Ben.
Ben Salmon from Oregon State University, our tech team on the Pacific Northwest.
Yes, hello everyone, good morning, or good afternoon, I guess, if you're on the East Coast. I'm Ben Salmon out of Oregon, and I'm going to talk about parasitic mite syndrome, otherwise known as PMS, which I think we all agree is not the best acronym, but we haven't really come to a consensus on the replacement term. I like VMS for varroa mite syndrome, but because there's also tracheal mites, which is a separate parasitic mite. But the dictionary definition of a syndrome is it's a condition with a set of associated symptoms. Symptoms and signs are often used interchangeably, but technically symptoms are experienced by you or me or a patient. That's a subjective term, and then signs are observed by someone else that are more objective and point to the disease or the syndrome. So this is a brood disease webinar, so I'm going to mostly be talking about the brood issues associated with mites, but just to review the signs of PMS in adults, or more colony-wide signs. Definitely you see mites on the workers and on drones. The colonies can get ornery and mad just because they're trying to fight the mites and they're not doing well. The colony will try to replace the queen. They think it's maybe a queen issue, so the colony will build supersedure cells. Also, the populations will shrink, and most commonly you'll see the deformed wing virus on the adult bees. They don't live that long usually, but usually if the mites are high enough, you'll see a good number of the workers with deformed wing. And we've got another poll here. I gave you some clues at that first slide.
We'll give you another 15 seconds. Okay, I'm closing the poll and I'm going to share the results.
You see all those, Ben?
Yes, so everyone pretty much on point. Pretty much all of those are signs of PMS except for the foul odor, unless you just have brood that's just rotting and dying, but that's typically not a sign of PMS. So going into the brood symptoms, there's signs you do see the shoot down brood where the workers are uncapping and actually starting to cannibalize the pupae normally, or they'll even start to, they'll suck up the larvae or just be picking at it a lot. The cells you'll start to see just, pinholes or openings in the cells where they're inspecting the brood, and they might be able to detect the mites or even detect distressed larvae, and they uncap it to check on them. Melted down larvae, some of the Foulbrood symptoms can look similar, but when all these are together, you can point to the mites as the culprit. Again, spotty pattern can be present in all these diseases. But one that I've actually noticed a lot of commercial beekeepers don't even realize or look for is the frass, the mite feces, which you can see on the far right picture there. That's a whole mite family that I got disrupted when I pulled the pupa out with the tweezers. But you can see the foundress, the mother mite, and her oldest daughter, which is also brown. And then the little male is the elongated one on top. And then there's a younger daughter mite, and then the egg below. But the frass is the white. It's very pure white material there on the ceiling of the cell. And I want to point out that you can see also in this picture the cocoon, which you don't want to confuse for mite frass. That's then the bottom of the cell, and there's a squiggly line with a little bit of yellow mixed in. The frass is always pure white, and it's on the ceiling of the cell as opposed to the bottom. Next slide, please. So the first thing I do open a colony is look at the brood pattern, and before I even start looking for the queen, I'm looking at the brood, and I'll tilt the frame up, and the mite frass can only be seen if you tilt the frame up and look at the ceilings of the cell. So here you can see with the red arrows that really distinct white material. Those were cells that had been opened and chewed down, the pupa removed, and they don't get around to cleaning that mite frass out right away. So you can see that pretty well. And then there in the bottom center, you can see that one is, you know, it's been chewed open and mite frass on the ceiling with a chewed down pupa. In the next slides, I have more of this mite frass. This one is a really obvious when you just look up at the ceilings. Almost half of those cells have mite frass on the ceiling. So the mite foundress, when she crawls into the cell, she goes into the top of the larvae and punctures an opening, starts to feed there, and then she defecates on the ceiling of the cell. And that's when she lays her eggs and those young mites are hatching, the young mites smell the feces and go up to the top of that, so they know where to feed on the larva. So that mite frass will always be in the ceiling. And here you can see a little closer. This colony was collapsing. The population was small. There were yellow jackets that were invading and robbing the honey and eating the larvae. So again, you can see the opening cells. They're being uncapped. And there's certain bees have a varroa-sensitive hygiene trait, which makes them very sensitive to the mites into the cell, and they'll obsessively uncap. That's a trait that queen breeders are trying to incorporate into their stock, so that those bees can disrupt the mite's life cycle. They don't actually kill the mite, but they'll uncap the brood, and that disrupts their life cycle enough so that that mother mite has to give up that family or abandon that cell, crawl out and try again in another cell. And after 2 or 3 tries, she's out of energy and usually can't reproduce. I think in the next slide, I even have a closer picture. Maybe not. This is going on to the other signs, this chewed down or melted brood. So at first glance, this could look like one of the Foulbroods, but in this case, there was tons of mites on the bees and deformed wings. So I knew that it wasn't probably a Foulbrood. But if you look closely, you can see some of them are pupae that were chewed down, but a lot of these were younger larvae that didn't make it to the pupal stage. So a couple things can be happening here. Either those larvae are malnourished because there's not enough nurse bees to take care of them. When the viruses get bad, the nurse bees, they're dying. They have deformed wing. They're not able to care for the brood. The queen is trying her best to lay and ramp up the population, but if there's not the nurse bees to take care of the larvae, they tend to die and they start to decay, and it'll give that appearance of the melted down look. And with the pictures from Rob and Dan, you'd see up close. You could see the with the EFB especially, you can see the trachea, the white lines. You won't see that for the melted down brood that's caused by PMS. Also, when the viruses get really high, the nurse bees can actually feed, they can actually feed virus to the larvae, and they'll die before they get to the pupal stage. And when the colony gets to this point, they're almost beyond repair. You need to be treating immediately, and even then, it's not guaranteed that it's going to make it. Okay, next slide. Here's some more up close pictures. You can see on the left, there's the mite right on the head of that pupa. It was recently chewed open, and the mite is going to crawl out and try to find another cell. And you can also see in the lower pictures the pupa. They don't actually pull it out. Sometimes they'll pull the whole pupa out, but if they can't do that, they'll just start to nibble on it and chew it down like that. So you'll see sometimes they'll be recycling that protein and actually cannibalizing that brood if they in need of the protein. And on the right there, you can see more of the melted down larvae that, when it starts to rot, there's bacteria that move in. It's not necessarily EFB or AFB bacteria, but other just decomposing bacteria will move in and start to rot the dead larvae, and they can look strange and look really gross and be all different colors, but it's just not necessarily a Foulbrood. But then if you look at the upper right of that picture on the right, you can see that worker with deformed wings and a mite right on its thorax. So seeing that melted down brood and deformed wing and mites in the bees, you pretty much know that it's a PMS situation here. So when I opened this colony, my first guess possibly was there was terrible mites and they were chewing it down, but you know, usually it's not this bad. So after doing the mite wash, I found 0 mites in the sample, and turns out that this colony was actually starving to death. So it was in the almonds. It had been left too late in the almonds after the bloom. There's really nothing for the bees to feed on in a lot of parts of California. So the bees need to be moved out soon or fed after the bloom is over. In this case, they were left to fend for themselves. The queen had ramped up when there was a good pollen flow and good nutrition, but then it just got shut off like a spigot, and the bees suddenly started to starve, and they just started chewing down their brood and eating it and to recycle that protein. So again, the spotty pattern in chew down brood can indicate a number of things, and it's just a sign that you have to look closer and determine what's actually going on. So if you have a 5% varroa level, that it's going to look a lot different depending on a lot of things, the time of year. If you have a 5% varroa level in the spring, that's like a death sentence for that colony. That's really high because most of the mites are under the cappings, and if you have 5% on the workers, you know, there's a lot more. Yeah, so the degree of population turnover. So yeah, and again in the spring, the queen is really ramping up. There's a lot of brood. The bees population can actually grow way faster than the mite population, so they can outrun the mites. Especially if the mites are starting low, they can outrun the mites for a while. But as soon as that brood production starts to plateau, the mites can really start to catch up, and that's why we see PMS most often in the fall when there's less brood and more mites. Also, the viral levels are really, the virus and mite interaction is really important. When mites first came on the scene in the 80s or 90s, I remember seeing them a lot in the 90s. You could sustain a huge mite population and the colony would still be able to pull through, and you'd see mites on the bees and the drones, everything, and they'd still be able to make it. But I think the viruses are really, they're changing, they're becoming more virulent and more deadly. And now, I mean, a 3% mite infestation is really troubling, because that's when the viruses, I think, really can cause great damage. And the viruses, it's normally what kills the colony, not the mites themselves. The bees were able to withstand it before the viruses got really bad. And also, like I was mentioning before, the hygienic bees, or the VSH colonies, will pick apart their brood a lot. So you might see that chewed down brood appearance and think that the mites are very high, but in fact, a lot of times I've looked at a few of these VSH colonies with very poor patterns, a lot of chewed down brood or opened caps, and the mite level is actually quite low because those bees are being vigilant about opening those cells that have mites and interrupting that life cycle. So a lot of beekeepers, they look at their hive, especially if they're not, you know, if they're just in the first few years of beekeeping, they won't see any mites, so they assume that they're fine. Their colonies don't have mites, and that's really a pretty common misconception. You really have to test for mites. So for every mite that you see on a bee, there's many more in the colony. Mites normally don't hang out on the backs of the bees. So that's where you're going to normally see them. On that middle picture, you can see the mite on the thorax. They're usually, if they're on the bees, they're almost always on the underside. They hang out on the bottom. In fact, when the bee hatches, a lot of times there's the daughter mites that are wedged under in the sclerites, the scales under the abdomen, and they'll be wedged in there feeding as that bee hatches. So one thing I like to do is, if I see some newly hatched bees, I'll just oftentimes just pick them up by their wings gently and look at the bottom. And if I suspect that colony has mites, if I see some deformed wing virus, I'll pick up those bees, and almost always, if a bee hatches with deformed wing virus, it has mites on them underneath. And as those mites mature, they'll drop off and start to crawl around. So really the best way to test for mites is the alcohol wash. That's what all of our field specialists do. Some people like to do the sugar roll or the sugar shake or the ether roll. Other methods of mite monitoring. We've done some trials on that, and it really, those other methods, they can give you an idea, and it's better than nothing, but really you can miss a lot of mites with those other methods. Alcohol wash, we get 95% or so of the mites off that way. And you're killing 300 bees usually, but usually it's a tiny fraction of the population and doesn't really make a huge difference in that colony. So we encourage everyone to monitor mites with alcohol wash throughout the season, and then depending on the time of year, the threshold, we say the threshold is 3 mites per 100 bees. In the springtime, I would think it should be even lower than that. If you have 3% in the early spring, like I said before, that means there's a lot more mites that are in the brood. So go to the next slide, please. Another poll. I think Net will set that up.
There it goes. All right, we'll give you about 30 seconds to complete this last poll. Is it running? I see 0 have voted, but it's running on my screen. Oh, here we go. There was a slight delay.
Just need to let them read that long question. Time.
I just did see a question about the percentage of alcohol that you need for alcohol wash. I think it's about 35% is the good amount of alcohol. So if you get the isopropyl alcohol or the rubbing alcohol from the pharmacy, that comes in 70%. You can just dilute that one part with 1 part water, and it'll bring it down to 35%.
Okay, we've got about half of the folks who have responded to the poll, so I'm going to end it now and share the results.
Yeah, most people did choose the right answer there. Wearing gloves, sure, it can. If you're changing the gloves between every colony, it can help prevent, especially if you see one of the contagious diseases. It would be a great idea to change your gloves if they're rubber gloves. But if you're wearing the gloves in the apiary, you're just, you're picking up stuff on the gloves and then going to the next, and it doesn't really do much to prevent the transmission of disease unless you're changing them often. We do wear the nitrile rubber gloves in the field, and as you can see in that picture there, and if I come across a colony that has a EFB or AFB and I'm messing around in that brood, I'll change those gloves out, and then also sterilize the hive tool. So to wrap up here, we'll just go through some of the prevention tips, the do's and don'ts. So practicing good hygiene is always a good idea. Keep your equipment clean. Use the torch to sterilize the hive tool. We sterilize between every beekeeper because we do visit a lot of beekeepers. We want to make sure we're not spreading any disease. So we sterilize everything, all of our equipment before we go to a new beekeeper and after ever seeing any kind of disease, the transmissible ones. So you can use the lighter for tweezers, or the torch. Alcohol works good as a disinfectant. Sometimes I'll be wrapping up in the yard and I'll toss my hive tool in the open smoker and stoke it up a little bit. Really melt all the wax off, flip it over, get the other side. That's just kind of a quick way to sterilize the hive tool. Close your operation. That just means, don't be buying junk from other beekeepers, and then isolate individual apiaries. Keep your apiaries somewhat spread apart, and don't swap equipment back and forth. Again, yeah, I'm kind of going into the don'ts already, but buying used equipment is how I think a lot of beekeepers end up getting the Foulbrood, EFB and AFB. That's a big problem. I think it's best just to buy new equipment. It might be a little bit more, but you'll thank yourself in the long term. And yeah, moving equipment between colonies or apiaries is a good way to spread the spore forming and bacteria diseases. And then robbing events. So as a colony gets smaller, if it's dwindling or dying, do something to that colony, either restart it or shake it out or something. Weak colonies will inevitably get robbed, and that's gonna, whatever disease that colony had or mites will be transferred to your healthy ones. So dealing with colonies before they get to that point where they're just dwindling to nothing is a good idea. And also, if you're opening the hives in the late in the fall or winter, or late in the summer or fall, just don't keep them open to encourage robbing. So once those bees start to rob, they don't stop. They'll just rob until they die. They'll get into that robbing mood, and it's really hard to stop it once it starts. And the last one, layout apiaries to promote drift. That's more of a tricky one. Some people think the horseshoe shape is best for that, or I just think it's, a layout that promotes drift is a huge holding yard where every colony is painted white, looks exactly the same. There's no landmarks. That's where we see the drift tends to be the worst. So with that, I'll wrap up my portion, and we'll go on to perhaps one last poll.
We don't have any more polls, but we do have a huge round of applause for all of our field specialists. Unfortunately, I know they can't hear you, but we can measure our applause here by the number of people. We hit our max at some point during this, which was 300. So we're looking to extend our capacity for future webinars, and also the activity in the chat box. We take that as your applause as well, because we've gotten so many good questions, and we're going to move into the Q&A in just a moment here. We are going to have another webinar in 2 weeks, so we will be sending out a note to this list, and we'll also post that on our social media channels and on our website. We're going to send the recording to everyone who registered for this. We know there were some people who were not able to get in because they did not log in within the 300, so we'll send that out. We do, as we showed in the very beginning, as we were waiting to start, we do have a disease manual that includes a lot of the photos that you saw in this presentation, and that you can check with your local bee suppliers, your favorite bee suppliers, maybe not local, but they carry it. We do have a note in the chat somewhere with a link for some of those. And then we are a nonprofit. We are feeling the impacts of COVID-19 just as all nonprofits are at this time. So if you can, please consider supporting BIP at our link below on this screen. And now I'd like to again thank our field specialists, Ben, Nelson, Rob, and Dan. They did an excellent job sharing this information, and we're going to open it up for questions. Eric will moderate our Q&A. If we don't get to them all, because we don't have much time left here, we will perhaps turn the rest of them into a blog. So if you're interested in that, go onto our website on the blog page, and at the bottom where it says sign up for updates, you can get on our blog. I also just want to mention, we do have a Sentinel program for those of you who are interested in help with sampling for mites during the season. Look into our Sentinel program, which is also on our site at beinformed.org. I'll stop there, and I'll turn it over to Eric, who's going to moderate our Q&A. Thank you.
Well, hello, everybody. Thanks so much again for joining us. I'm not going to take a lot of time to say much other than go ahead and get right into the questions. We've got about 12 minutes for this. So we're going to start with some general questions. Adele Towers was asking if it would be possible to get the PDF slides after the presentation. And I guess I'll defer to Anne-Marie on that one. Anne-Marie, do you know if that will be made available?
Can I speak to that? This is Nat. I will say that we do have the recording available, and that will be posted. I'm not sure if we are going to provide the slides in a format yet. We'll need to consider that, but we will definitely have access to the recording.
Excellent. Thank you so much. And again, presenters, amazing job. That was remarkably informative. So thank you again. A lot of people in the chat were applauding you there as we were getting out. So next question. There was a question by Ari Benjaminson. He was asking if we could talk about the national distribution of the various diseases. I don't think we've got time to get into that, but if anybody wants to take a quick stab at it, tech teams.
I'll take a stab at that. I don't know. I think all these diseases are present all across the country at this point. I don't think there's any part of the country that's protected or for whatever reason has avoided these problems. I do see more variation though in pollination crops. It seems like certain when bees are in certain pollination crops, you'll see certain problems crop up more. Blueberries come to mind. I feel like I see EFB in the colonies that are in blueberries almost every time. Also, it depends on the quality of the forage. In certain locations, you just have poor forage. The Central Valley of California is not that great of a place for most of the year. Also, certain agricultural environments where there's just a lot of intensive ag, corn and soy. Those locations where the bees aren't getting good forage, they just need a little extra help, and they're more susceptible, I think, to some of the diseases in general.
Gotcha. Okay, thank you very much. I appreciate that. Next question. Again, this is for anybody on the team who knows. There's quite a few comments asking about alcohol being hard to find. They're wondering if we could use windshield washer fluid, if that would need to be full strength or diluted. Also, if we could use denatured alcohol for mite watches. And any tech team member can just chime on in.
Yeah, as it relates to using a windshield washer fluid, yeah, we use alcohol for all of our mite washes, and I've had to switch to windshield washer fluid, but you want to make sure that it's the winterized windshield washer fluid that has the concentration of alcohol. I think it's 30% that we're currently using right now, windshield washer fluid, with the 30% concentration of alcohol.
Okay, and also just be careful when you're handling that, because you do need to read the label, because it of course wasn't intended for that purpose. It was just meant to be poured right in. And if you've read the warning instructions, you do not want to get that on your skin or in your eyes. So just if you are doing that, be careful. Next, we've got a question from Daryl G. It's kind of another general question about decontaminating equipment. How long should you leave the tool in the alcohol if you're using alcohol to decontaminate that?
I'm not really totally confident on this answer, but I would say just to dip it and swish it around should be enough to kill anything on it. Anybody else?
The main thing is you want to remove any wax, honey, propolis, things like that, and then rinse. So scrub it first and then rinse it with alcohol, bleach.
Yeah, good point.
Excellent. Thanks, guys. All right. So, and I also know that Dr. Megan Milbreth recommends using a bleach solution to, again, treat your equipment between yards, so disinfecting. Okay, so on to a couple of AFB questions from NR. At what point should you not even treat and just throw everything away?
What did you say that was for, Eric?
That's for AFB, regarding AFB
Okay.
specifically. Yeah.
With an infested colony, I dispose of it and treat the yard regardless of the level of infestation. I'm also inclined to burn all the woodware, the boxes. Say you can sterilize them, but I don't DIY sterilization of equipment in that scenario, I would rather burn a box for $12 than potentially reinfest a new colony. So that's how I approach it.
Gotcha. Cool. Thank you. Also, actually, Dan Wines, a couple of questions for you. Kathy Clay asked, my neighbor told me they had a hive come down with AFB. I'm super worried. What can I do to prevent any spread to my hives? We are within a mile from each other.
Yeah, the joys of beekeeping. Your animals interact with your neighbor's animals. Not a lot you can do aside from encouraging your neighbor to have dealt with it swiftly and appropriately. You know, so your bees or other bees can't rob it out, but there's not a lot you can do to restrict your bees from interacting with that colony if it's left alive and dwindles and dies.
Gotcha.
I also encourage your neighbor to really confirm that it is AFB by using a test kit, because we do see people assuming they have AFB when it's actually something else. So just make sure that it is indeed AFB before you start to worry that much.
And we thank you guys. Thank you very much. And we did get a number of questions about the Holst milk test as far as reliability. And you know, do you guys, can you speak on that briefly?
Yeah, I put a link further up the chat to it.
Okay.
I think it's reasonably reliable. It's not something I have a lot of firsthand experience with. I've found the ropey test to be adequate occasionally with these little field tests that we mentioned. But it is a viable method if you want to carry powdered milk and little test tubes around. That's, you know, that's perfectly acceptable way to go about it.
Gotcha. Cool. Thank you. So we've got a number of questions about the sanitization of for AFB, but I think the majority of that was answered. Somebody did ask about if using a hot wax dip is a way that you could sanitize your boxes.
Yes, I believe it's a function of temperature and time, duration above a certain temperature. I believe it's 10 minutes at 160 Celsius.
Gotcha. Okay, cool. Thank you. Okay, so we're going to move on to some other related questions for other diseases, just because we don't want to focus solely on AFB. But Dan, thank you so much, and that's amazing. So EFB question. So we've got a couple here. What are some concerns that are there with reusing supplies with EFB? And I can't remember. I think that was Rob.
So if I bought used equipment, I would probably just monitor those hives at least for a year or more, just to see if anything pops up. And that's what I would do, or even put that equipment in an isolated yard away from your current colonies, so you won't affect them if they do have it. But isolation and monitoring are your best ways to just make sure that no diseases pop up. And I would do that with any of the diseases. With new equipment, I would just put them in an isolated yard, so you don't have to worry about contaminating your newer stuff or your already previously bought equipment.
Can I chime in here? Rob, can you tell us if you have a colony that you know died or dwindled of EFB, how do you handle the equipment that was contaminated with EFB previously?
Ideally, I would try to use the equipment right away and treat the colonies as if they had it. I don't think storing it would do much as far as getting rid of it. Could try freezing the frames, but I don't know the efficacy on freezing in European Foulbrood. I don't know many people that are freezing frames that have had the disease. They usually just reuse the equipment and
Gotcha. Cool. Thank you. So we're gonna one more question for the EFB. Just to and if you've covered this, please let me know. But how does the microorganism spread in the hive if it does not produce spores? Is it by contact? and that's from Laura Vilmar.
So it's basically in contaminated brood food, and then it's fed to the nurse bees. That slide, I did forget to take the spores out. I kind of took that slide from Dan's AFB slide and kind of modified it a little bit, and it made me say the wrong thing. So yeah, it shouldn't say spores on there because it's spread through the bacteria in the brood food, and then the nurse bees spread it to the larva that way.
Okay, very cool. Thank you. So next is a Chalkbrood question. We only have about a minute left, so anything, we're going to try to address these in a later blog post, I believe Net mentioned. We have a lot of questions still, but just one for Chalkbrood, Nelson. If there was a question of, do screened bottom boards help prevent Chalkbrood?
Yes, that would align with increasing the ventilation within the colony, increasing that airflow. So yeah, I think especially in the springtime, it would be beneficial to have screened bottom boards on your colonies, not only for Varroa, but for as a hopefully prevention for Chalkbrood.
Excellent. Cool. Thank you. I didn't know that. So, Net, I do want to ask you, we do have a couple more questions about PMS. Can we go over a little bit if people are not, Lisa Stinson's really insisting on information about the donkey?
Okay, let's do one more question. I'm just afraid that Zoom might cut us off, not that we can't go on, but if we'll do one more, and then all the follow-up ones we will, or all the other ones, we will follow up with a blog post of some sort answering the majority of those.
Okay, wonderful. So I'm gonna go ahead and jump to this last question for the PMS, and this would be for Ben. Ben, I guess if you see deformed bees or deformed wing bees, what can you do for the hive? Or is it, I guess, is it needed? I'm not quite, there's a typo in there, but that's okay. So is there anything you can do for the hive that would help improve the situation, I guess?
Well, you can't really help the bees, of course, that have deformed wing. They're goners. They're not going to live for very long. But first thing I would do is, you might just want to buy your mite treatment right then and there. I mean, when you see deformed wing, that means the mites are already too high. So you should have been probably treating before you see that. But I would make sure you have the mite treatment on hand, and I would actually do a mite wash or test for your mites right away to see where they're at. But yeah, the deformed wing is just an indication that the mites are present and they're causing problems, and you should be on it pretty soon.
Gotcha. Cool. Thank you. And then there was one more question. Roxanne Crosby asked, should the hive be requeened if the bees become aggressive in the presence of parasitic mites, or does aggression subside after treatment?
Yeah, in that case, I mean, the mites are just causing the bees to be antsy and angry, and I would think that as soon as you get the mites under control, normally the colony will settle down and the queens fall to the genetics. Gotcha.
And I think we'll just do the one more question. And actually, this one just came in, and I saw it pop up earlier. Beth is asking, what is the best mite treatment? Enjoy.
That's opening a whole can of worms there, but there's a bunch of them on the market that work fine. Certain ones work at certain times of year better. Apivar works pretty well in the spring. So the thymol-based ones, I think, have a good place. Oxalic acid and formic acid are both organic acids that have good success with those. Some of the HopGuard, the newer formulations are satisfactory. So I'm probably missing some, but there are plenty of treatments out there. You just have to look at the time of year and your situation and what's available. Anybody else have any comments about treatments?
Excellent, thank you. And Dan Wines just shared a link in the chat as well. So I think that's pretty much it for the number of kind of unique questions. I mean, we do have more, but I think that those we can address at a later time.
Thank you so much, Eric, for moderating. And thank you again to our presenters. Really appreciate you guys taking the time to give us all this information. And like I said, we did record this, so we'll post this on our website and also send an email to all who registered. Join us in a couple weeks. We'll announce that one soon. And again, thank you for your time and have a great afternoon. Stay healthy, everyone.